Diseases of ginger. First is leaf spot, that is caused by Phyllostickta gingiberry. Initial symptoms: water-soaked white spots on the leaves surrounded by brown margins, yellow halo, adjacent lesions enlarge and coalesce to form necrotic areas. So leaf spot of ginger is caused by Phyllostickta gingiberry. Initial water-soaked white spots on the leaves surrounded by brown margins. Yellow halo. Adjacent lesions enlarge and coalesce to form necrotic areas. Spray copper oxychloride at the rate 0.25% or mancozeb at the rate 0.2%. Leaf spot of ginger is caused by Phyllostickta gingiberry. Then soft rot or rhizome rot is caused by Pythium aphanidermatum or Pythium vexans or Pythium myriotylum or Fusarium solani. So soft rot or rhizome rot of ginger is caused by 4 types of species or 4 types of fungal pathogens. First is Pythium aphanidermatum or Pythium vexans or Pythium myriotylum or Fusarium solani. Pythium aphanidermatum, Pythium vexans or Pythium myriotylum or Fusarium solani. Soft rot or rhizome rot of ginger. Yellowing first on the lower leaf surface, then upper leaves. Drooping, withering and drying of pseudostems. Drooping, withering and drying of pseudostem. Affected rhizome produce roots. Rotting of roots. Brown discoloration of rhizome tissues. First, yellowing of lower leaf surface, then upper leaves. Drooping, withering and drying of pseudostem. Affected rhizome produce roots. Rotting of roots. Brown discoloration of rhizome tissues. Rhizome treatment with 1% Bodox mixture and again with Trichoderma viridi at the rate 8 to 10 gram per liter. Plants drenching with 0.2% of copper oxychloride or Bodox mixture. Bacterial wilt. Bacterial wilt is caused by Ralstonia solanacearum. Sorry. Bacterial wilt in ginger is caused by Ralstonia solanacearum. Enlarged water-soaked patches or streaks on the collar region. Bronze discoloration of leaf margins. Leaves droop down, typical wilt symptom. At advanced stage, slimy pseudostem. If you press the infected rhizome, oozes out a milky bacterial exudate in clear water. Bacterial wilt. So bacterial wilt in ginger is caused by Ralstonia solanacearum. Enlarged water-soaked patches or streaks on the collar region. Bronze discoloration of leaf margins. Leaves droop down, typical wilt symptom. At advanced stages, slimy pseudostem. If you press the infected rhizome, oozes out a milky bacterial exudate in clear water. Rhizome treatment with 30 minutes. Rhizome treatment with streptocycline at the rate 200 ppm and shade dried before planting for 30 minutes. Drenching with 1% of Bodox mixture or 0.2% of copper oxychloride. So which type of antibiotic is used against bacterial wilt of ginger? That is streptocycline. Rhizome treatment. Yellow disease or wilt of ginger is caused by Fusarium oxysporum forma specialis gingiberry. Yellow wilt. Yellow disease or yellow wilt of ginger. Thread blight by Pellicularia filamentosa. Bacterial wilt, Pseudomonas solanacearum. Mosaic disease by wheat streak mosaic virus. Root knot nematode Melidogyne incognita. Yellow disease or yellow wilt of ginger is caused by Fusarium oxysporum forma specialis gingiberry. Thread blight by Pellicularia filamentosa. Bacterial wilt by Pseudomonas solanacearum. Mosaic disease by wheat streak mosaic virus, which is the virus of mosaic disease in case of ginger, wheat streak mosaic virus, and the root knot nematode in ginger is Melidogyne incognita. Then rhizome rot or soft rot have aseptate mycelium. Asexual spores are zoospores. Sporangiophore encysted. Sporangiophore produce zoospores. Sporangium. Sexual spores are oospores and oogonium. Vegetative spores are chlamydospores. PSI oospores, chlamydospores in primary inoculum. Primary source of inoculum is oospores and chlamydospores in infected parts. Secondary source of inoculum, zoospores through irrigation water. Tolerant varieties, Vengara, Vinard local. Rhizome rot or soft rot of ginger. Aseptate mycelium. Asexual spores are sporangium produces sporangiophores produces sporangium and zoospores. Sexual spores are oospores and oogonium, and vegetative spores are chlamydospores. Aseptate mycelium. Which type of mycelium? Aseptate. Primary source of inoculum oospores and chlamydospores in infected parts. Secondary source of inoculum zoospores through irrigation water. Tolerant varieties Vengana, Vinard local. Vengana and Vinard local of ginger. Then leaf spot. Leaf spot is caused by Coletotrichum gingiberis. Leaf spot of ginger is caused by Coletotrichum gingiberis. Primary source of inoculum dormant mycelia. Secondary source of inoculum airborne conidia. Gametangial contact acarvules, ascospores, perithecia, conidial discharge. So leaf spot of ginger is caused by Coletotrichum gingiberis. Primary source of inoculum dormant mycelia and secondary source of inoculum airborne conidia. Gametangial contact is happened in the sexual spores. Acarvules, ascospores, perithecia, conidial discharge. Phylosticta leaf spot. Pycnidiospores remain viable for 14 months. Phylosticta leaf spores, pycnidiospores remain viable for 14 months. Yellow disease or wilt. Phylosticta leaf spot. Pycnidiospores remain viable for 14 months. Yellow disease or wilt. Yellow disease or wilt is caused by Fusarium oxysporum forma specialis gingiberri. Primary source of inoculum chlamydospores and perithecia. Secondary source of inoculum soil-borne and airborne macro and microconidia. Primary source of inoculum in yellow disease or wilt, chlamydospores and perithecia. Secondary source of inoculum soil-borne and airborne macro and microconidia. Epidemiology acidic pH, clay loam soil, soil application of neem cake pine needles, increased native population of Trichoderma species. Epidemiology acidic pH, clay loam soil, soil application of neem cake pine needles, increased native population of Trichoderma species. Thread blight of ginger is caused by Pericularia filamentosa. Leaves lose their turgidity. Bacterial, in case of thread blight, Pericularia filamentosa in ginger, leaves lose their turgidity. Bacterial wilt etiology, gram-negative rod-shaped motile bacteria. Epidemiology sandy loam soil. Primary source of inoculum is infected rhizomes and many weeds, and a secondary source of inoculum irrigation water. Single polar flagellum. Bacterial wilt in ginger is caused by Ralstonia solanacearum or Pseudomonas solanacearum. Etiology gram-negative rod-shaped motile bacteria, single polar flagellum or monotrichous, gram-negative rod-shaped motile bacteria. Primary source of inoculum is infected rhizome and many seeds. Secondary source of inoculum irrigation water. Epidemiology is sandy loam soil. Sandy loam soil is epidemiology. Root knot nematode is Melidogyne incognita. Female nematodes present in the root galls. Secondary source of infection is or inoculum second stage juvenile. Neutral soil pH is the epidemiology. Fourth juvenile stages are there. Mosaic disease of ginger. Secondary source of inoculum is Viruliferous vector. Root knot nematode Melidogyne incognita. Female nematodes present in the root galls. Secondary source of inoculum second stage juvenile. Neutral soil pH is the epidemiology. Four juvenile phases are there in the root knot nematodes. Mosaic diseases of ginger. Secondary source of inoculum is the Viruliferous vector. Mosaic diseases, wheat streak mosaic virus.